Okay, now let's talk a little bit about RNA. Okay, so there are three main types of RNA. There's mRNA, tRNA, and rRNA. The one you are most familiar with is mRNA, and it's the coding RNA, which is translated to proteins. However, tRNA and rRNA also have important non-coding functions in the cell. rRNA is called ribosomal RNA, and it makes up many important parts of the ribosome, which is where protein translation occurs. The ribosome is essentially the machine that that basically turns RNA into proteins. So rRNA is a component of the ribosome, right? It's an important component. The details you don't really have to know of what this is, but just know that it's a component of RNA, of ribosomal RNA. tRNA is called transfer RNA, and it has a very important function in translation. Its function is to bring the correct amino acid to the ribosome for translation. As you know, each three-letter mRNA sequence called a codon corresponds to an amino acid. Like, for example, CUU corresponds to leucine. Each floating tRNA is bound to a specific amino acid as well. So the tRNA is actually bound to an amino acid, and it brings the amino acid into the ribosome. For example, the anticodon for leucine would be AAG. Remember, codons are always written 5 to 3. 5 to 3 prime. So the thing for CUU, 5 to 3, would be AAG. And this is because they're anti-complementary, so the G and the C, the A and the A, and the A and the A, right? So if you want to see it like this way, try and write upside down. Right, so this is 5, 3, 5, 3. So the anticodon for CUU would be AAG. The cell knows this, and it knows that, okay, if I know that if in the DNA it's written CUU, or the RNA it's written CUU, I need to put a leucine onto it. How do I know CUU is written there? Well, I'll try AAG. If I bring an AAG to this CUU and they bind perfectly, that means there's a CUU there. If there's, for example, a AUG, and then you bring an AAG, it won't bind perfectly, so it knows, okay, leucine doesn't go here because this doesn't bind perfectly. But if this happens, it brings an AAG here, and it binds perfectly, the cell knows, okay, there's a CUU there. There's a CUU there, I have to give it a leucine. So this anticodon is attached to leucine. So it knows, okay, this is a perfect match. So that means leucine goes here. So it takes this leucine and adds it to the growing polypeptide chain, which I'll just write like this. Okay, and that's the protein, like the growing protein is being made. So if it notices that the anticodon and codon have a perfect connection, it knows, okay, the codon is coding for the amino acid that this specific anticodon is bound to. So that means take this protein, this amino acid off this anticodon and move it here. And this anticodon is called the tRNA, the transfer RNA, because it literally transfers the amino acid from just the outside space into the ribosome and allows it to move onto the growing peptide chain. Okay. All right. Going off of that, let's talk about degenerate code and wobble pairing. Remember that our mRNA is degenerate, meaning that there are actually more codon sequences possible than amino acids. Thus, there are multiple sequences that code for the exact same amino acid. This is due to something called wobble pairing, where the anticodon and codon can have a mismatch in the codon's 3' end and still pair normally. Give an example. And this is actually a good image. So this is the tRNA. And you can see this tRNA is bound to an amino acid leucine right here. This whole thing is like the tRNA amino acid complex. And it has, on this end, it has this anticodon GAG. And notice how here it has GAG, CUC. Normal pairing, it knows, okay, leucine goes onto this amino acid chain. Leucine goes here, basically. Whereas here, it's CUU, but this is JAG. It wouldn't be a perfect match because this needs to be a or this needs to be a C for it to be a perfect match. But there's this thing called wobble pairing, which will still say, okay, it's close enough, that's fine. It matches close enough. Still, take this leucine, put it here, put it on the chain, even though it's not a perfect pair. This only works on the 3' end of the codon of the mRNA, or the 5' end. Remember, this is 5' to 3', or the 5' of the anticodon. 5 of the anticodon or 3' of the codon for wobble pairing. Only this. And this is the exact reason that both CUC and CUU both code for leucine. This is the reason that that's allowed to happen. Two different things can code for the same amino acid because of this wobble pairing. Because both of them can pair with this tRNA that has leucine attached to it. So for example, if you had a problem, and this is a very MCAT-like question maybe, if you had a mutation where this tRNA, GAG, instead of being bound to leucine, there was a mutation that bound it instead to the amino acid glycine. Whenever you would have a leucine in the, if you have a DNA sequence, wherever the DNA sequence told you to put leucine, it would instead put glycine, because it would say, okay, well, the pairs match, GAG matches with CUU. Okay, take the amino acid that's bound here, put it on the growing amino acid chain. But it wouldn't put leucine like it's supposed to, it would instead put glycine. Okay, so that's a very MCAT-like question that they would ask. Like, what would happen if you just took every anticodon or tRNA that's supposed to be bound to leucine, replaced with glycine? What would happen to the protein that it was made? All the proteins that are supposed to be leucine would be changed to glycine. Hopefully that makes sense. Okay, next let's talk about exons and introns. As mRNA is the most commonly seen RNA on the MCAT, you should know not only its function in translation, but also how it's modified post-transcription. In prokaryotes, all of the DNA is converted to mRNA, and all of the mRNA is translated into protein. Very simple. Eukaryotes, it's not true, it's a little more complicated. Once all of the DNA has been transcribed to mRNA, the mRNA is then processed, and the unneeded introns are removed from the sequence by the spliceosome, which is another machine, leaving only the exons for translation. And it kind of takes, so you had exon, I'll share it here. Go here. You have X, int, X, and just keep going. Takes the intron, cuts it out, and then it kind of glues these two together. And so it just goes X, X, these two sequences, and just puts them all together essentially. Right? Takes out all the introns and then glues the exons together. That's the job of the spliceosome. Exons are usually shorter in length and they contain unique sequences, while introns are usually much longer, but they contain simple and repeating sequences. Most of our DNA, sorry, this mistake was to say our, most of our DNA is introns due to their length because they're much longer. We have a lot more DNA that's just introns than exons. So that's why most of our DNA actually isn't ever translated, or it's not supposed to be translated. It's just there, okay? And you may think, well, if we're just have that DNA just to not be translated, what's the purpose of having it? This is a very highly studied question by many researchers, because having introns requires much investment by our body. Mainly, it requires the organism to have and maintain spliceosomes which are big complexes and they require a lot of materials to make them and to keep them functioning and running. And having introns also increases the risk of mutations, because if you have a mutation in the introns, you could have something where it accidentally changes the introns so that they don't get spliced out and they end up being translated, and that makes a completely different protein than what was supposed to happen. So it increases the risk of dangerous mutations. And there's no visible evolutionary benefit. However, it has been noted in research that the presence of introns greatly affects the expression of exons. So for example, if you take all the introns out of an organism that's supposed to have introns, all of the exons will be expressed much less than they would normally be. And that's because introns, some known functions of the introns is helping initiate and terminate transcription. It also adds alternative splicing, which allows a greater number of gene functions to be encoded in a smaller genome, and I'll talk about this in a little bit. But essentially what alternate splicing is, I mentioned there's exon, int, x, whatever. And this is like this part, this has like this is long, then another exon's like this. Normally these two would be, this would be cut out and these two would be glued together. Alternative splicing means maybe instead of doing that, say if you had another intron here, maybe you could just cut it out in a different way, and you could do like this, and then like this part as well could be joined into there. Essentially, you can kind of like mix. You can have multiple combinations within just one DNA sequence of introns and exons of what's needed and not needed, because introns, they allow you to kind of create different combinations with the same genomic sequence, and those different combinations would obviously produce completely different proteins, which would have different functions. So you kind of save space by having not just two whole different sequences back to back to create two different proteins. You could have one big sequence that can be cut this way, or it can be cut this way to make a certain protein. Okay. But again, one of the main functions of introns is we're not exactly like fully sure why, other than they help initiate and terminate transcription. It really helps to have to increase gene expression. So genes that are more commonly expressed. So we've seen in research that genes that are more commonly expressed in our bodies than genes that aren't more expressed have more intron sequences than genes that aren't commonly expressed. So we know that introns are very important to increase overall gene expression. But the exact full details aren't clear, but this is like some of the known stuff. And again, there's a lot of research going on about it. Okay, lastly, let's talk about the spliceosome. The spliceosome is a large complex found in cells that is the machine responsible for intron splicing. It is comprised of a mixture of RNA and proteins. The RNA in this complex is called snRNA, which functions in base pairing with the pre-mRNA post-transcription. Again, once the pre-mRNA comes out of transcription, it gets modified. So when the DNA comes out of the transcription, it base pairs with the snRNA, and that kind of allows it to go through the transcription that it needs, the post-transcriptional modification that it needs to. It's made up of molecules called small nuclear ribonucleoprotein particles, or snRNPs, very long name, just abbreviated to snRNPs, which are protein RNA complexes that join together to make the spliceosome complex. Okay, so they're snRNPs, which are protein RNA complexes that are small. They each have their own small function, but a bunch of them join together to make the spliceosome complex. And it functions by recognizing certain sequences in the pre-mRNA called intron splicing sites, which tell the complex where the introns are and where it should cut. Okay. All right. So that's it.